Hi, I'm Caitlin, and this summer I was working with some lovely researchers at the Bioeconomy Science Institute out in Lincoln on a project exploring the diversity within a New Zealand collection of Xanthomonas arboricola pruni, which is also referred to as ZAP. ZAP is a plant pathogen that causes bacterial spot disease in stone fruit. It can affect crops by reducing fruit quality, lowering yield, and just overall impacting their marketability. Although ZAP is recognized as a very important plant pathogen, there isn't a huge amount known about how much variation exists within New Zealand isolates of ZAP, or whether that variation translates into meaningful biological differences. So the overall goal of my project was to look at the diversity within this New Zealand isolate collection and link the genetic diversity with two practical outcomes: the antimicrobial susceptibility and host pathogen interactions. So, in other words, I wanted to look at variation on DNA and how it actually translates to disease-related behavior. To address that, I used a combination of approaches. First, I examined the genetic relationships among isolates by comparing the New Zealand collection with reference sequences. And then I tested antimicrobial susceptibility, focusing on copper and streptomycin because of their relevance to the disease management in fields. I also screened for known resistance-associated genes to see whether any tolerance patterns could be explained genetically. And I also investigated host interactions, seeing if any isolates originally from one host species behave differently when infecting other host species. There were three main findings of my project. Firstly, most isolates were very genetically similar. The majority clustered with ZAP reference sequences, suggesting relatively low divergence across this collection. Second, there was no evidence of widespread antimicrobial resistance in this collection. Most isolates were highly susceptible to copper and streptomycin, and we did not detect common resistance-associated genes at all. This suggests that the established resistance is not widespread in this collection, bar one isolate that showed highly elevated streptomycin tolerance, which is particularly interesting as a target for some follow-up work. And thirdly, the phenotypic variation still did matter. Even though most isolates were genetically very similar, disease severity differed depending on the particular host and strain combination. So we originally looked at this to see if we could find evidence of host specificity, where one isolate, say, for example, isolated off of an apricot, would only be able to infect other apricot species. We did not find evidence of this. They were able to effectively inoculate across different hosts. However, we did find host-dependent behavior, meaning isolates interacted differently with different host species when they did infect, in terms of like disease severity and such. So together, these findings suggest that the population is broadly quite genetically conserved, mostly susceptible to antimicrobials, but still has biologically relevant variation in disease expression, which makes it useful as a baseline for future monitoring, particularly for the emergence of divergent or antimicrobial tolerance under continued field use. Thank you, especially to everyone who supervised me at the Bioeconomy Science Institute in Lincoln.
Thank you, Lynn.
Thanks.
Questions? Peter.
That was fantastic. Awesome. I don't think I've ever said this, but what an attractive bacteria that looks like on the place. It's really pretty.
It is pretty.
How long, can I ask how long each of these strains have been in culture? Is this something that has come out of plants recently and then you're characterizing, or is this culture collection been sitting in BSI and being grown on plates for 20 years?
These strains were initially taken approximately a decade ago, mostly from the Hawke's Bay region, as well as some Canterbury sampling sites, as a part of someone's PhD originally, my supervisor. And so a large part of why we wanted to do this work is because we wanted to establish a baseline for the diversity of these strains 10 years ago, particularly so that we can go out and do fresh work and compare over the last decade continued antimicrobial pressures being put onto these strains, what might have changed. So in a way, we wanted to do this as a background, as a basis, but no, the strains were not freshly isolated.
Cool. Thank you. Very
Yes.
nice. Thanks.
Got time for another question? Ian.
I totally agree. That's a beautiful bacterium and a good name, ZAP. My question was, you didn't find any genetic variation, but you found those phenotypic variations among strains. Do you have
Yeah.
any ideas about how that's happening?
No, it is definitely an interesting one. I think the main thing here is like genes not being detected doesn't necessarily mean that a resistance mechanism is not there. A major limitation, of course, is that we're only screening for genes that we know are associated with resistance mechanisms. There could be other underlying alternative mechanisms, mutation-based resistance, altered uptake. The strain that we did see this behavior in did we did observe that behavior across all replications of the test for that isolate. So basically, the next step is to try and work out what is different about that isolate if it's not genetically different or not significantly genetically different on the basis we looked at.